In this video, we are going to look at the science behind carbon dating. This is a method of determining the age of an object containing organic material by using the properties of a radioactive isotope of carbon. Carbon dating is used to date objects like wood, plant remains, charcoal, animal parts like bone, leather, horn, and many others. To understand carbon dating, we need to understand the forms of carbon. The forms or types of carbon are mainly three. We have carbon 12, carbon 13, and carbon 14. When you look at carbon 12, this is the most common isotope of carbon. These three are known as isotopes. However, carbon 12 and carbon 14 will be the backbone of this discussion. The first two, carbon 12 and carbon 13, are actually stable and as such will not easily break down. However, carbon 14 is unstable due to the extra 2 neutrons present. It can easily break down to form a new element, and this will actually be the secret behind radioactivity and carbon dating. So how is carbon 14 produced and taken up into the ecosystem? The energy coming to Earth as cosmic rays can cause carbon 14 atoms to be produced. So these cosmic rays can interact with the atmosphere, producing neutrons in the due process. If a neutron of the right range of energy collides with a nitrogen atom, a reaction takes place. So when you look at our nitrogen, it can collide with a neutron to produce our carbon 14, and this is how carbon 14 comes into existence, with the emission of a proton. This is our proton. So when this carbon 14 is produced, it will be incorporated into carbon dioxide on oxidation, and then this will take the path of photosynthesis in the presence of water, and then our carbon 14 will be stored as starch, carbohydrates, proteins, and so on, as long as carbon 14 has been used in our carbon dioxide. So since animals feed on plants as well, and fellow animals can feed on other animals, even we human beings can take in carbon 14. Before we look at how carbon 14 decays, let us assume that the Earth is bombarded at a constant rate with cosmic rays. In this case, the rate of production of carbon 14 should be constant, and as such the ratio of carbon 14 to carbon 12 must also be constant. So when living organisms die, obviously the carbon 14 will have to decay and change to nitrogen 14 through beta decay. So our fossils, the bones, the wood, the horns, will give off certain emissions that we shall call beta particles. So our carbon 14 will actually undergo beta decay to form back the nitrogen 14. Carbon 14 will decay back to nitrogen 14 with emission of a beta particle, which is our high-speed electron. So one thing to note is that in all living organisms, you realize that always the ratio of carbon 12 to carbon 14 should be a constant. So in this image here, our ratio of carbon 12 to carbon 14 is 2 to 1, because organisms are constantly taking in carbon 14, and this carbon 14 is gradually being converted back to nitrogen 14. So at some point equilibrium is established. So the ratio of carbon 12 to carbon 14 is always kept constant. However, when these living organisms die, obviously the ratio will have to change, because carbon 12 is not decaying, it's not breaking down. All atoms will be present, all atoms of carbon 12. However, when you look at carbon 14 gradually decreases, and as such the ratio starts to change. As seen, here the 3 atoms of carbon-14 are no longer present. However, they have been converted into atoms of nitrogen. So in a dead material, the ratio of carbon-12 to carbon-14 gradually changes, and this is the basis with which man uses to determine the age of these different samples. If we are to compare the amount of carbon-14 remaining with time in terms of number of half-lives, in this case R100 will mean here the organism is still alive and all carbon-14 atoms are present. So our graph will have to start from 100. However, as time goes by, when this organism passes on or it dies, what happens is that the carbon-14 will start gradually decaying into nitrogen-14, as we have seen. So what happens is that carbon-14 has what we call half-life, and its half-life is 5,730 years. That means if you have 100 grams of carbon-14, after 5,730 years, you will be left with 50 grams of carbon-14, with the other 50 having converted to nitrogen-14. So when you look at our graph here, this is the actual graph of carbon-14 decay, in such that after each half-life, after every 5,730 years, you realize that the amount remaining is always half the initial amount. So if the initial amount is 100, after one half-life, the amount is 50. After the second half-life, another 5,730 years, the amount will be 25, and so on. So if you find your sample contains maybe 75% of carbon-14 remaining, you can have a graph of this sort plotted, and then you just extrapolate and see on your time scale how many years will this sample have passed on or have last lived. So you can use such a graph to determine the age of that given sample. How carbon-14 from the sample is measured. This is where the real dating starts from. There are many methods. 3 are the most common. However, in this video we are going to look at 2. Radiometric dating. This one involves radiations. Man tries to use machines to detect emissions given off by that given sample whose age is to be determined. So radiations given off by carbon-14 have to be noted in what we shall call activity. So the activity of that sample is noted through radiometric dating, and somehow they can use graphs to detect how old that given sample is. However, we also have the accelerator mass spectrometry, also known as AMS, and this one uses mass. And just like you hear spectrometry, basically the machine tries to sort out the different isotopes of carbon basing on their masses, because remember, they have different masses because they have different mass numbers. So all these are separated in terms of masses, and man can be able to determine how many atoms of carbon-14 are present and how many atoms of carbon-12 are present. So using the theory that for a living organism, carbon-14 to carbon-12 ratio is constant, man can be able to work backwards and determine the age of that given sample. However, there is serious preparation needed in all these methods, things like cleaning the sample, things like heating, sometimes turning them to graphite, and so on. However, when we look at calculating the age of a sample numerically or mathematically, we basically use equations. We can use this equation here. This equation here can be modified depending on how good your mathematics is. So we have natural log of n naught over nt times is equal to K times T. So when you look at N naught, this can be in terms of activity if you're using radiometric dating, or in terms of number of particles if you're using the AMS, number of carbon-14 particles or atoms, sorry. So number of carbon-14 atoms, that is when the organism was still alive, when time is equal to 0. However, NT will be the number of carbon-14 atoms after some time. So this will be got from the accelerator mass spectrometer. The machine can give you how many atoms of carbon-14 are present in the sample. However, the value of K is known as the decay constant. So the decay constant of carbon-14 can be obtained from its half-life. And when you look at half-life, is given by 0.693, also be written as natural log of 2 over K. So we can get the K because we know the half-life of carbon-14, which is 5,730 years. So once we obtain K, we can obtain the number of atoms of carbon present in that sample through the AMS machine, and then we can also get the actual number of carbon-14 atoms, because we know that the ratio of carbon-12 to carbon-14 is always a constant. As such, we can always find the time, T, in terms of years in most cases. However, it also has its own error, so sometimes plus or minus maybe 20 years. So this is how we can determine the age of a given sample. However, it's much more complex than it is theoretically. What do you think are the limitations of carbon dating? You know, we might believe this is the only way. Sometimes scientists have to combine it with other dating methods like the tree ring dating and others, to see whether their estimations concerning the age or the date of that specimen are within correct values. So let me know, what do you think would be the limitations of carbon dating, or how effective or efficient is it to date different samples? Thank you for watching.